All right. Hi, everybody. Thanks for being here today. My name is David Heiser, and I'm the Director of Student Programs at the Yale Peabody Museum. And it is my pleasure to welcome you to today's Research Spotlight Talk. This is a series that we started now about a year ago that features graduate students from Yale who are doing work on Peabody collections or related topics. We're nearing the end of their time as a graduate student and have something really interesting to share. You may have already noticed, but if not, live captioning is available, and you can find that by going to the bottom of your Zoom window, and you can select to turn the captioning on. We've turned the chat off, but if you have questions or comments, please use the Q&A function, which is also found on the bottom bar. And that's how you can communicate with us today. So I'm really pleased today to introduce today's Research Spotlight speaker, Morgan Moeglein. Morgan's in the final stages of her graduate career here at Yale, and she just handed in her dissertation this past Monday. So way to go, Morgan. Hey, good to see you. All right, so Morgan is originally from Alaska, where all her family is, and decided that she wanted to go to college somewhere really far from Alaska. So she chose University of Vermont. So that's, yeah, that's quite a distance. I had a great experience there studying biology and really got into plant biology at the University of Vermont. And did some field work in Ecuador, did some collecting. Then after graduation, moved back to Alaska for a while and worked as a gardener. So still, a deep-seated interest in plants clearly had developed at UVM, if not before, but then moved back to New England, worked at MIT as a lab manager for a while, and then decided she wanted to take her career to the next level. So applied to a graduate program at Brown to work in Erica Edwards' lab. After 3 years, Erica was hired by Yale, which is how we got Morgan. Morgan moved with Erica here to Yale and finished her doctorate here, or is almost finished in the process, and is doing great work that she's going to tell you all about. She is, in addition to the work that she's going to tell you about, she's also really curious about the development within buds of plants and thinking about how plants invest in tissues that are sort of risky tissues, as she puts it, these plant tissues that have to somehow make it through the winter as they do within buds. So I think that's a postdoctoral work direction for her, but today she's going to be telling us about the work that she's been doing in the Edwards lab for her dissertation. She's coming to us today from Boston, Massachusetts. And with that, I just want to welcome Morgan Moeglein. Thanks, Morgan.
Thanks. Okay, I'll go ahead and share my screen.
Great.
Sorry, 1 second. It's not quite working the same way.
Huh, no kidding. All right.
There it is.
All right, great.
Okay,
right.
Can you see and hear everything? Okay, great.
Yes.
So thanks everyone for coming on this really, really nice day. I'm sure you might rather be outside, so thanks for joining us. So today I'm going to be talking about my dissertation work, especially the parts of my dissertation where I did a lot of fieldwork and research on some plants in Mexico. So plant leaves are highly variable, and leaf trait differences can be diagnostic of species differences as well. So if you've ever tried to identify a plant and you've opened your plant identification book, you've probably seen a plate similar to this one. So this is one of my favorite tree ID books, and if you open to one of the first pages, it tells you about different leaf traits that might be useful in your species identification. So on the top right, you can see all the ways that that leaf shape might vary. Leaves might be small and elongate. They might be large and round. Also, leaf bases and leaf tips can be diagnostic. They can be really variable. And also on the bottom right, there's these diagrams of leaf margins. So margin type can be really useful in distinguishing species, and you'll get everything from these entire margins that are just smooth along the edges of the leaf to these toothy margins where they might be kind of jagged or rigid, all the way up to these lobed margins that are more like a maple or an oak tree. They might have these indents along the side of the leaf. So there's a lot of leaf trait variation, and botanists have been really fascinated by this for hundreds of years, just going out, observing differences in the field. And it leads you to wonder, what might the purpose of all of this leaf trait variation be? And our understanding of why leaves might be so variable drives partially from our understanding of how leaves work and leaf physiology. So leaves are the primary site of photosynthesis for most plants. They need to take energy from the sun and convert it to sugars so that they can live, they can power their cells, they can go on growing. And this is the formula for photosynthesis. So in order for photosynthesis to occur, the leaf needs to take in CO2 from the atmosphere. And it also needs water. So that's water that gets pulled up from the soil and moves through the plant. And it also needs energy from the sun to power this process. And as a result, you get sugars. So that's the C6H12O6. It lets the plant survive. It lets the cells function. And then it also produces oxygen as a byproduct. So in each leaf, it's this constant balance between pulling in CO2, having enough water to power this equation, and then it's also losing oxygen out of its leaves at the same time. So when you think about water movement through a plant, the water is coming up from the roots, it's moving through the stem, and then it gets into the leaf. And the water is moving through the veins of the leaves, but then it eventually can exit the plant through these little pores in the leaf called the stomata. So this picture on the bottom right is a picture of a cleared leaf specimen. So the chlorophyll has been cleared out and it's been stained so that you can see the individual cells. And these little pores in the leaf surface, these little orange ovals, these are the stomata. So they can open and close, and they take in CO2 from the atmosphere. So that's that first part of the photosynthesis equation. And in the process, oxygen and water also diffuse out. So this is the site of what we call gas exchange. The CO2, oxygen, vapor water, they're all diffusing through these stomata. And how a leaf configures its veins, its stomata, its overall shape is going to have an effect on how successful it can be at performing photosynthesis in a variety of different environments. So one way that leaf traits are related to leaf function is through this thing called the boundary layer. So over any surface, you have, in this case, a leaf, you have this envelope of air, this boundary layer, where when moving air moves over the object, if the boundary layer is thicker, air moving over it is going to move more slowly and in a more irregular pattern. It's not just going to fly right over that object. So if a leaf has a thicker boundary layer, it's going to have disrupted laminar airflow. So air moving over it is not going to move as quickly or as smoothly. And if you're a leaf and you are taking in CO2 and losing water vapor and oxygen, this thicker boundary layer, this little envelope, can slow your gas exchange. So the diffusion of gas is going to just happen at a lower rate. And this little insulative envelope of the boundary layer is also going to hold temperature. So a leaf is going to be slower to equilibrate to air temperature if it's kind of insulated by this boundary layer space. And so different leaf shapes and leaf qualities can have implications for boundary layer dynamics. So if a leaf or any object really is larger, then airflow over it is going to have more of a chance to slow down. So it's going to have a thicker boundary layer as opposed to a smaller leaf, which would have a thinner boundary layer. This also matters in terms of margin characters. So if you have a leaf with an entire margin versus a leaf of a similar size, but that is toothy along the margin or maybe lobed, that toothy or lobed leaf is going to have a thinner boundary layer because it has more little spaces along the margin. It has effectively a smaller size where air can move between the teeth much more quickly. And finally, the leaf surface is also going to matter for boundary layer dynamics. So if you have a very hairy leaf, if your leaf is pubescent is the botanical term for it, it's covered in these little hairs, these trichomes, that's going to create this rougher leaf surface where when air moves over the surface of the leaf, it's going to be more slowed down. There's going to be this thicker boundary layer effect in comparison to a leaf that is glabrous or not hairy. So there are a lot of leaf trait differences across plants, but there can also be really pronounced differences within smaller groups of plants. And this can be kind of a more manageable size of number of species to work with. And so we are particularly interested in leaf trait diversity within Viburnum. So all of these leaves here are leaves that were cleared and stained from different species of Viburnum. And you can see that they're super variable. So there's a lot of different shapes here, a lot of different margin characters. On the bottom left, we have a nice entire margin. And then on the top right, we have this finely serrated toothed margin. Some of these are also lobed, so some look a little more like an oak leaf or a maple leaf. We really get a lot of leaf trait diversity within this smaller group, similar to what we see across plants in general. And another benefit of working within a smaller group is we might have a better understanding of evolutionary relationships within and among the species. So in this case, for Viburnum, we have a pretty well-resolved phylogeny. So that is this diagram on the left. And there's all these little species names to the right side of the phylogeny. So there's about 165 species total. And the lines on the left side of the phylogeny is sort of a family tree showing how these different species are related to each other. So which species are more closely related, which ones are more distantly related, when did they branch apart over evolutionary history? And that can help us have a better understanding of how these leaf traits might have evolved in the group. So today we're just going to focus in on one little clade within the Viburnum, the Arenatinus. And this is a group of species that has radiated down through Central and into South America through these high elevation cloud forest environments. And looking specifically at species within this radiation, we are going to try to answer two different questions. So first, are species with similar leaf traits found in similar environments? Based on what I just told you about leaf physiology and boundary layers, we might have certain expectations based on morphology as to what kind of environments we might find this in. And if different species have the same morphology, we might expect to find them in the same kinds of places. And then after that, we're going to ask, are some leaf traits more likely to be found together because they are genetically linked? So related to this idea that maybe environment is selecting for certain groups of leaf traits, An alternative hypothesis could be that we find certain leaf traits together because they are genetically linked. So maybe they're controlled by the same parts of the genome, or maybe they're controlled by genes that are very proximal to each other, very close together. So that is also an alternative and a potential explanation for why we see the leaf traits that we do. So getting into the first question, it kind of stemmed from our observations in the field, going back to some early trips from Michael Donahue in the 80s, where he was traveling through Mexico and just trying to get a sense of these species and their differences. So what I'm going to talk about today is kind of focusing on 3 different areas in Mexico. First, the area around Puebla, so that's the orange dots in the top left of the map. Some areas around Oaxaca, so those are the blue dots, and then also Chiapas, so these purple dots to the bottom right. And in each of these regions, we find these recurring leaf syndromes. So one of these syndromes is our large pubescent and toothed leaves. These are big, hairy leaves, and they tend to have these toothy margins. And we can find different species in different regions. So for example, Viburnum microcarpum on the top left, we only find that in Puebla. Viburnum sulcatum and new species 1, we find those in Oaxaca. And Jucundum we find in Chiapas. And going on with some other syndromes we find, in addition to our large hairy toothy species, we find these small glabrous, so that's not hairy, leaves with these entire margins. And again, we find a different species in each region that fits this syndrome. And then finally, we have a sort of funny intermediate syndrome as well, these large glabrous entire species, where they have these bigger kind of elongate leaves with these smooth margins and very few to no hairs. And when we go back to their phylogenetic relationships, it's really interesting. So we have different little clades within this group that are endemic to different regions. So we have a group of closely related species in Puebla, another group of closely related species in Oaxaca, and another group of closely related species in Chiapas. And in each of those regions, we have the independent evolution of these different syndromes. So over and over again, we have, hairiness, toothiness, and large blade size coming together, and et cetera, for all of our different syndromes. So this was really cool, and we wanted to get a better understanding of if maybe environment was, selecting for these leaf syndromes over and over again. And so the first thing we really needed to do was just explore where these are living, get a better sense of it. I ultimately made 9 trips down to Mexico over the course of 3 years, just kind of getting a better sense of these places. And the environmental differences are really subtle. So they all grow in these high elevation cloud forest environments. So these are pretty wet environments, high elevation. And in the picture on the right, you can see that there's kind of this fog rolling in, these clouds. And that's really typical of this environment, thus cloud forest, where a lot of the moisture environment is really determined by water brought in by these daily cloud banks. And so after we identified or felt we had a good sense of where these different species were living, we installed these climate stations to get a good sense of the microclimate within each species patch. So climate stations were installed in all these locations on the left, where there's these little colored dots in each region. And on the right is our climate station. So it consists of a data logger, so that's that white box, a temperature sensor, so that's kind of that white beehive looking thing. And then attached to the temperature sensor is a leaf wetness sensor. So it's a little thin blade of plastic that's meant to approximate how wet a leaf might be. And this is really important for a cloud forest environment because mistiness is really important to measure when understanding water relations in these plants. In addition to these, we also had soil moisture probes under the ground to give us a better sense of rainfall and soil water relations at the root level. And so our first clue that maybe this wasn't as clear as we expected was when we just looked straight at elevation of our different leaf types. So we kind of thought, if our different leaf syndromes are sorting by the same environmental cues, maybe they would be sorting by the same elevations. So each dot here is a population of one of our species, and they're colored by their different syndromes. So the green ones are big, hairy, toothy species, purple dots are small, glabrous, entire, and yellow dots are large, glabrous, entire. And you can see that at each region, they're pretty well mixed up. So they're not following the same elevational sorting across sites. And this is really important because, as you can imagine, elevation has a really strong correlation with temperature in general. So as you move up in elevation, it gets colder. And that is what we see in our climate station data. So each bar here is a site representing the average temperature at that site. They're all colored by leaf type. And then the bars at the bottom are colored by region. And so within region, temperature goes down as elevation goes up, but there isn't sorting for leaf syndromes based on temperature. However, when we looked at diurnal temperature change, so that is the difference between the highest temperature in a given day and the lowest temperature, we did start to see some signals. So especially in our small glabrous entire leaves, those are the purple bars, we tended to see a bit more diurnal temperature change at those sites than we necessarily did at other leaf syndrome sites. And specifically, those sites were way more likely to encounter freezing. So that's designated by the little blue snowflakes. So all of our small glabrous entire sites encountered freezing, while only one site that was not small glabrous entire encountered freezing. And this is kind of consistent with our boundary layer dynamics, again, where for these small leaves, they can equilibrate with air temperature really quickly. They can heat up, they can cool down. So these little forays into freezing, which might be detrimental for a bigger leaf, might be more tolerable when you have this smaller blade size. Then when we looked at moisture environment across our sites, we found another surprise. So we found that our big, hairy, toothy leaves were really occupying two different extremes in the moisture environment. So our large pubescent toothed species are these green bars, and this is the number of wet leaf minutes per day. So that's the number of minutes that our leaf wetness sensor has some mist on it. And you can see that the green bars are either the highest values or the lowest values. And so it seems that even though we have this syndrome where the leaves seem really similar, they're actually bifurcating into these two really different moisture regimes. And we think this might have something to do with the ways that these species are hairy. So even though they're all hairy, our dry hairy species tend to have these really, really dense hairs, as you can see on the picture on the bottom left, while our wet hairy species hairs are a little bit more diffuse. And we think it may be that these very dense hairs could serve to keep a leaf from drying out. So if they're covering the stomata, it might slow that diffusion of water out of the leaf surface as maybe an adaptation to drought, while our wet hairy species may be serving as, the trichomes may be serving as more of a little umbrella to kind of keep water from inundating the leaf at all times. So are species with similar leaf traits found in similar environments? Sometimes. However, it's not necessarily too clear. So even though we have different species within a same syndrome, they're not always doing the same thing. And it should still be noted that there is a lot of environmental overlap. Our stations were able to find these fine differences, but at the same time, these are all found along similar cloud forest environments. So it might not be such a strong determinant in determining leaf syndromes. So then, given that maybe environment wasn't the whole story, we wanted to know, are some leaf traits more likely to be found together because they are genetically linked? If it's not the environment bringing these syndromes together over and over again, maybe there's something about these different traits where they're more likely to be inherited together because of genetic reasons. So to look at that, we went back to our phylogeny and we found these two species who are sister species with very divergent leaf types. So sister species means that they're each other's closest relative. These species did not diverge very long ago. They're very closely related. They're genetically pretty similar, but they have way different leaf types. So Viburnum laudum has these small, glabrous, entire margin leaf types. It's a close-up of the leaf blade in these bottom pictures. And Viburnum jucundum on the right, you can see is quite hairy. It has these really branchy hairs on both sides of the leaf surface. These two are really different from each other. And when we explored across their ranges in Chiapas, we actually found that they were able to hybridize pretty readily. So we found quite a few environments, especially disturbed areas like along roadsides, where these two were still able to cross, and their resulting offspring had all these different leaf phenotypes. So you have some leaves that are small but toothy, you have some big leaves with entire margins, and kind of everything in between. And when we quantified this variation, it really appeared that, at least phenotypically, these traits can be broken up and reshuffled and put back together, so they must not be so tightly genetically linked together. But we wanted to know more about the underlying genetic variation behind these syndromes. So what we did next is we subsequenced the genomes of all of our hybrid swarm individuals, along with some of our parental species, Viburnum laudum and Viburnum jucundum. And what we found was a nice genetic gradation between the two. So the mauve bars represent the proportion of the genome associated with laudum ancestry, and the teal bars represent the proportion of the genome associated with jucundum ancestry. And you can see that a lot of our hybrid swarm individuals in the middle are very genetically mixed. And we found that if we plotted genetic ancestry by our leaf traits, there was a strong correlation between ancestry and phenotypes associated with a parent species. So in this case, jucundum ancestry on the x-axis is correlated with blade area or trichome density on the y-axis. We then wanted to know if there were specific little regions of the genome that were most strongly associated with our traits of interest. So in these plots, every dot is a region of the genome that is different between one parent species or the other parent species. And if a dot is higher up in the graph, it means it's more strongly associated or correlated with our trait. So in this case, our traits are blade area and marginal teeth, so the teeth along the edge of the leaf. And blade area had a lot of different regions of the genome that had a really strong signal for association for increased blade size. While marginal teeth, there weren't as many regions of the genome associated, but there's two regions, these two points that I've circled, that are associated with both traits. So it appears that even though there might be lots of ways to increase your blade area, a couple of those are also associated with becoming more toothy. We also looked for genetic correlations with trichome density on the abaxial and adaxial side of the leaf, so that's the top and the bottom. And we found that there was also one region of the genome that was associated with hairiness on both sides of the leaf, meaning that if you're hairy on one side, it might be more likely that you would also be hairy on the other side of the leaf, but it's not a perfect relationship. Interestingly, there were also no regions of the genome that were associated with blade area and teeth, and also with hairiness. So those seem to be able to vary independently of each other. So in summary, it seems that these traits can be dissociated, even though we find these syndromes across Mexican viburnum and Arenotinus in general. It does seem possible, at least from our hybrids, that genetically these traits can be broken apart. However, there are some associations between traits, at least for blade area and marginal teeth. So in conclusion, there do seem to be some climatic variables selecting for leaf type, but it doesn't explain necessarily all the syndromes we find. And not all climatic variables seem to be as important as others. There also seems to be a genetic basis for leaf trait, leaf trait evolution here, and traits can mostly vary independently from each other, but there are some correlations. So given this, it seems that leaf trait syndromes may be held together by some selective force not yet identified, even though climate and certain genetic markers might be kind of nudging these syndromes, these over evolutionary time into these syndromes. So I would like to thank everyone from the Edwards Lab. This is a collage that Erica started making a really long time ago, and I love that it just represents all the people that have directly or indirectly worked on this project over time. And everyone at Yale and Brown EEB, who I've worked with over my doctoral work. Everyone at UNAM, so down in Mexico, there's this great university we work with who enables our fieldwork, including Mark Olson on the right of that selfie, and the National Science Foundation for funding this work. Thank you.
Thank you, Morgan. Fantastic. That's super interesting work. I didn't reveal earlier, but my own background is in botany, and so I'm particularly interested in this story. And yeah, it's really neat to think about Viburnum as a genus having such incredible diversity in leaf shape and you know, these other elements of the leaves, the margin, the hairiness, that sort of thing. I mean, it's really just fascinating to think about why that might have happened, and cool that you delved into that. So I encourage people to put questions in the Q&A. I know there are already some in there, but I want to start with one that is, I mean, my guess is it's probably one that other people are asking themselves. And I hate to put you on the spot right away with this, but so you mentioned in your last slide, there may be other selective forces at play here that have not been yet identified, but I have a feeling that you've at least thought about what some of those might be that could be the topic of further research in this area, or maybe you and your lab group have talked about what other selective forces could possibly be driving these kind of patterns that you see. So any conjecture about what might be likely to be you know, also at the cause of this?
Yeah, there can be lots of conjecture. Of course, over the course of a dissertation, you just can't even come close to testing everything. I think one really important thing to keep in mind is, even though we see these leaf syndromes that might seem, you know, super obvious to us and really interesting, they exist in the context of, you know, a whole plant. So aside from having these different leaf shapes and different, you know, trichome densities and stuff like that, these plants can be entirely different sizes. So plant height, these can be shrubs, they can be small trees, sometimes fairly large trees. So that sort of dynamic of like what role they play in the canopy within their given ecosystem could be super important. We've observed a lot of differences in flowering time in the field. So we generally think of these as kind of generalist pollinators, maybe bird dispersed, but we haven't like really quantified that. So those could be some interesting differences in terms of dispersal, in terms of things that might correlate with flowering time that we wouldn't even think of. And then there's things we wanted to test that we just never got to, like soil substrates, super important. Soils can be really hard to work on because it's not something our lab necessarily does a lot of, and the permitting can be challenging, but yeah, and then just, you know, hydraulic physiology across the plant outside of the leaves as well. So yeah, there's a lot of other ways that they vary beyond just the leaves, for sure.
All right. So there's more work ahead in this area, it sounds like. So yeah, I was particularly taken by your slide of the cleared and stained leaves, and because in that slide you could so clearly see the correlation between the veins of the leaves and their margins. So when there were lobes, you could see a big vein going into that lobe, and you could see veins going into the teeth. And so you kind of get a sense that these lobes and teeth follow the venation of the leaves. And that sort of coupled with this notion that you were telling me before about your interest in studying, plant investment in tissues. Anytime a leaf, is formed in a plant, that requires energy for the plant. And evolutionarily speaking, on any given plant, the shape of that leaf represents some kind of evolutionary trade-off between investment in the development of those leaf tissues and other sort of energetic factors or physiological factors of the plant, which made me think more about the soil and the sort of kind of nutrient regime that these plants might be up against, and wondering if there's any evidence, not just in Viburnum, but in general, that somehow with toothiness or lobiness, if you will, that somehow represents a balance between the plant sort of losing a little bit of leaf tissue in the missing spaces between those lobes, in order to compensate for some kind of maybe lower nutrients where it evolved. Is there any work that's been done on that?
Yeah, that makes me think of a lot of different things. So, so first off, I know this is the case for Viburnum, and I think it's the case across a lot of other woody species in general, and something I'm hoping to do more of in my postdoc is when you look at how these leaves are built from the really tiny stages, investment in veins is usually, in terms of like bulk mass, seems to be what's built first. So if you go out to a tree in the winter and you pull a bud off of it and you start dissecting it, the little tiny leaves that you'll see inside of that bud, in my experience, they tend to be like all vein. And something I've always wondered about is if they're just making really tiny cells that they just haven't inflated yet, or if they are actually just making vein cells. And then in terms of your point on lobiness and toothiness and investment, I'm not so sure about nutritively what the trade-off might be exactly, but there is some evidence from earlier work, and then also some papers that Erica Edwards and Michael Donoghue have written, where there's this idea that if things are more lobed or toothed, it might actually, they might be able to pack more efficiently into buds. So when you get rid of that tissue between the veins, it can make it easier to kind of fold things more efficiently into a little space. Yeah. So as a trade-off overwintering kind of thing, there might even be an advantage to not having that tissue. But that's something where more work to just better understand that is ongoing.
Great, thank you. So yeah, let's go to some questions here. How did you choose what leaf traits to study? And I think that question comes probably from your very first slide where you showed that hairiness, margin, and size were not the only traits that you could have chosen. So how did you choose?
Yeah, so it was kind of informed by patterns that we saw in the field, but then also the broader literature. So there's this study from 1916 where people, Bailey and Sinnott, went out and just collected leaves from all over the world, and they found that leaves with toothier margins were found closer to the poles more often, while if you looked at plants closer to the equator, they tended to have more entire margins. And there's been more and more work with that over the last 100-something years that's kind of recapitulated that there tends to be toothier margins in cold environments. And even within individual plants, if you grow, clones at different temperatures, the colder clone can often end up with toothier margins as well. There's a cool study showing that in maples. So toothiness has this long history of this really interesting connection to climate and environment. And again, like why, there's been a lot of hypotheses, but it's not like definitively proven, and it might be different from plant to plant. So toothiness was something, and toothiness was also something that in previous Viburnum work, they had kind of found that pattern again across the viburnums of Asia, where as you went north, the viburnum leaves tended to be toothier, and as you went south, they tended to be more entire margined. So that was a trait that we were already really keyed in on because of previous work and previous understanding of it. And then blade size and hairiness were just in the field, just like so obviously different that we thought something might be going on with this, especially between our small glabrous entire type and our large hairy toothy type. And especially when we looked at sister species that were divergent, it was just a really big change over brief evolutionary time. So that made us think there must be some sort of like strong selective pressure to really drive a wedge between those two species. And hairiness as well in these cloud forest environments. We just really wondered, the trichome literature is super conflicting as to whether trichomes are catching water. Some plants can actually do what's called foliar uptake. And this is something that is, I think, more common in cloud forest environments where it can actually pull that misty water through the leaf surface. Wow. So yeah, there's a lot of intriguing literature on trichome dynamics, and we wanted to better understand that, how that worked in there.
Well, that was actually exactly one of our other questions, which was someone who wondered whether hairs help collect fog moisture in dry areas and if there's any difference in the effectiveness of various hair types for that. So you've basically just said there probably is.
Yeah, it's not definitive, but there's a lot of cool evidence for just you can kind of adapt trichomes to do all these different things. They're not always necessarily doing the same thing in different places. So I feel like there's so much more work to do on trichomes.
I love plant hairs. I remember in a botany class in college, a professor saying, yeah, if you think mammals have hair, you haven't seen anything. So yeah, I encourage anyone on this webinar to put a leaf under a microscope sometime and check out the hairs. They're really remarkable and really varied. So yeah, and the fact that you're looking at whether maybe the quality of the hairiness could differ enough, to be a factor in the diversity that you see. I think that's really cool too. So maybe you initially started off just thinking that hairiness was a little bit more sort of a yes or no character, but then ultimately teasing apart the fact that it might be a much more complicated story than that. That's cool. We had another question here. I'm wondering about the importance of the degree of toothiness. Do very shallow teeth have much effect on boundary layer dynamics? Maybe the yes-no split is obscuring some of the correlations.
Is that my advisor?
It is not your advisor, but it is. I happen to know a plant biologist who is writing this.
Yeah, no, that's a really good question. I think that teeth are definitely more pronounced in some of our species than in others. And the teeth are definitely more pronounced in kind of close relatives of the Arenotinus radiation. So when you get more into Viburnum dentatum or closely related things that are in North America, I think that's actually maybe where the depth of the teeth might matter more. But yeah, I think it was something that like we saw was different among our species, but how much it actually made a difference, we didn't actually test.
All right, fair enough. Thanks. One final question, because it's about 3:45 and we're going to close up here with just one more. Someone asks, why did you not measure leaf surface temperature? Or maybe you did, but you didn't talk about it. So yeah, did you measure leaf surface temperature?
Yeah, that would have been a great idea, but really hard to execute in the field. So, I mean, we could have gotten a very good snapshot in the moment when we went down and checked our stations. But installing these climate stations was, in a long-term way, was really, really difficult. They got vandalized, they got torn down. The more above ground parts that were visible, the more likely it was to get destroyed. So if we had some sort of sensor on a leaf surface, I worry it just would have been a little bit sensitive to destruction. But that would definitely be a better metric. Like that would be more accurate for sensing for sure.
Yeah, I mean, I think that the work that you did, that correlation with sort of freezing is really a compelling one, and so yeah, one wonders if you had a really finer scale look at the temperature, if that might have been able to be, if that might have been illustrative in some way. So
Yeah, we were really surprised at how often sites encountered freezing at all, because none of these are deciduous, so they hold on to their leaves year round, and some of them will still have, a couple weeks out of the year where they dip into freezing, which seems like it would be tough.
yeah,
So we were surprised that we found as much freezing as we did.
well, very cool. Thank you, Morgan, so much for sharing your research with us. Really, really appreciate it. I also want to just thank Megan Sullivan, who was behind the scenes on the Peabody team, fielding questions. Chris Renton, also behind the scenes on the Peabody team, and our captioner for great live captions today. So if you are interested in learning when things like this are happening again, our next research spotlight, perhaps we have a science cafe coming up next week. All of our programming, you should follow us on social media. You can go to our website, and there's a box on the homepage that says connect, and you can click on that and sign up to receive our emails. We want you to know when we're doing things like this, but mostly we appreciate you being here today. And Morgan, I just want to take this opportunity to say thanks again to you.
Yeah, thanks for having me.
Absolutely. All right, take care, everyone.